guess I'm just a play date to you.
Hello everyone, today I'm gonna do a presentation about meiosis. So let's begin. Many organisms pass their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information onto their own offspring. Then gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have two copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only one copy have of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germ cell. Similar to mitosis, the cells also pass through the interphase G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are two cell division events during meiosis. The first division, Meiosis 1, results in two unique daughter cells that have half the amount of DNA as the parent germline cells. The second division, which is Meiosis 2, results in four unique haploid cells that only have one copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Now, here we see the duplicated sister chromatids join together at the centromere, and then they stay fused at the centromere through meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving two pairs of sister chromatids. Chromosomal material is exchanged between the two pairs of sister chromatids. This event is called recombination, or more commonly crossing over, and we'll speak more about crossing over at the end of the video. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no two siblings aside from twins are genetically identical. The nuclear membrane begins to break down. Then the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are Telophase One and Cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2. Again, chromosomes condense the nuclear envelope and breaks down and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is that in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Why? Because since the sister chromatids are no longer identical. There will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cycle are pushed further apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. Since meiosis 2 began with two cells and each of those cells were split into two cells, and two gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. This resulting embryo then grows through many cycles of mitosis. And now we continue to move on explaining what is crossing over and genetic variation. So what is crossing over? So crossing over is during prophase 1 of meiosis, a part of chromatid of one chromosome goes over to a chromatid of other chromosome and vice versa. This interchange of the parts of the chromatids of a pair of homologous chromosomes is known as crossing over. Crossing over is a process by which two chromosomes of homologous pair exchange equal segments with each other. Crossing over also occurs in the first division of meiosis. So what's the importance of crossing over? It produces new individuals having new combinations of traits. It helps in establishing the concept of linear arrangement of genes. It helps in mapping of the chromosomes and selection of a useful recombination. And now we go on what is genetic variation, and we will explain lastly the law of independent assortment. So let's begin with genetic variation. During meiosis, homologous chromosomes, one from each parent, appear along their length. The chromosomes cross over the points called chiasma. At each chiasma, the chromosomes break and rejoin, trading some of their genes, and this recombination results in genetic variation. So now let me go and explain the law of independent assortment. The law of independent assortment describes how different genes independently separate from one another when reproductive cells develop. So during meiosis, the pairs of homologous chromosomes are divided in half to form haploid cells. And this separation or assortment of homologous chromosomes is random. And now I have most of the important facts about meiosis, and then that's it. So firstly, meiosis, remember that it occurs in reproductive cells, and number of chromosomes is halved in the daughter cells. And the daughter cells are genetically different to the parent cells and each other. Four daughter cells are formed, and homologous chromosomes come together. And lastly, there is exchange of genetic material between chromosomes. And now we have come to an end. I wish you have enjoyed the video. And by now, I'm sure that everyone has understood what is meiosis and the full explanation of meiosis. And thank you.